Good afternoon. My name is Dave Peden, and as you heard, I'm an allergist, and I direct the UNC Center for Environmental Medicine, Asthma, and Lung Biology. In addition to our EPA funding, we also carry on any given year about $5 million from the National Institutes of Health and a variety of grants, the vast majority of which are to either understand the impact of air pollution or environmental agents on worsenings of asthma. What I hope to convey to you today is a sense of, some of this you will know, but a sense of, a little bit of where ozone comes from, what ozone actually does to human beings when you inhale it, and how it might specifically worsen things for people with asthma. To the extent that there's time, it's more your time than mine. If you have questions during the course of this, I'm happy to entertain those then. I will be here for a little while afterwards if you have questions for me then. So with that, let's move ahead. I usually do one of these slides just so you'll know some of the other agencies that I'm involved with. This is more germane if I'm speaking about a specific medical intervention, but those are some of the other activities I have that disclose those activities. So as I said, what I hope to do is to give you a sense of the mechanisms or the ways in which ozone modulates or worsens exacerbation of environmental asthma or asthma caused by environmental agents. I also want to talk about how oxidant stress, how many of you have heard about oxidant stress and a specific gene that actually occurs in a lot of people that may be a risk factor for this. The potential for antioxidant interventions that could be used, vitamins or other therapies that might be used to specifically for people who are at higher risk for asthma exacerbations due to ozone, and things that we as a society can do, and some actual tangible examples that societal interventions do have an effect on health outcomes. There are a couple of facts that I think are really, if you don't take away much of anything else, there are a couple of key facts that I think are really important to know because it will help you understand when you would expect worsenings of health to occur in the context of an ozone exposure. Generally, increases in asthma exacerbation occur 24 to 48 hours after the pollutant exposure. And that's actually true not just for ozone, but that's true for particulates and other air pollutants as well. Many people hear the ozone alert and they're expecting to see immediate worsenings of asthma that day. And typically, what most of the data show is it's the following day where the problem is going to occur. In actual fact, for the summer months, many of the same kinds of things that or atmospheric events that would lead to increased ozone are also the same things that lead to heat strokes. So it's actually, if you're involved with healthcare for student athletes, it's the same kinds of events that occur. And in fact, it's the mid to late afternoon that's probably the risk period for ozone exposure because that's when ozone is typically at its peak. The other thing is that exacerbations have been shown to occur with pollutant exposures that are less than the current national ambient air quality standards. How many of you have know what the national ambient air quality standards are? Okay, well, it's a kind of an arcane acronym, but basically those are a variety of atmospheric pollutants, and those are the standards that by the Clean Air Act represent the national standard at which air pollutants really should not rise above. And they are routinely reviewed by law by the administrator of the EPA. And some of the kind of background slides I saw flashing before this talk spoke to the fact that the ozone standard is about to change. And the reason is that actually fairly low levels of ozone for perhaps a prolonged period of time can worsen things. With regards to asthma, and specifically regards to asthma in people who are allergic to things. So how many of you know someone who has asthma? No. All right then. How many people have asthma? Okay. How many people have allergy associated with their asthma? Okay. So in children, 80 to 85% of children with asthma have allergy as a significant, if not the leading risk factor for development of disease. If you develop asthma as an adult, that number drops to about 40 or 50%. And there are some other mechanisms that lead to the same kind of irritation that causes asthma. But what asthma is a chronic inflammatory disease of the airways. So, and the kinds of chronic inflammation you have are with a bunch of cells called eosinophils, and they're cleverly named because a microbiology stain called eosin is what lights them up. They look orange if they're done right. If I did them, they don't look like anything. But a person who knows how to do the staining, it'll look reddish in the slide, and they are cousins to the white blood cells that fight bacterial infection, and they cause chronic irritation of the airway. That chronic irritation of the airway is associated with increased responsiveness by a person with that inflammation to all kinds of things, whether you're allergic to them or not. So many of the actual triggers of an acute asthma worsening does not really occur because you suddenly are, I mean, it can occur if you're allergic to cats and you go to, Aunt Millie's house and Aunt Millie's got 4 cats. How many of you own cats? I've offended about a third of the room. But, so a sudden acute exposure to allergy or allergens, things that you're allergic to, can worsen asthma, and I think that might be important in ozone effects, but in fact, other non-allergic things can cause that as well. So a general concept is that the chronic allergic inflammation is what really sets you up to respond to all kinds of things. If you worsen that inflammation, you're more prone to things later on down the road, and the two things can work, both allergic and non-allergic irritants can work together to cause acute worsenings of disease. Okay. These are the atmospheric agents that are currently regulated by the Clean Air Act, and they're relatively recent standards. The ones that are in green, which in my original version of this was red, but as I've transformed from one platform to the other, they turn green. And actually, this standard, the 0.08 part per million ozone standard, which is an 8-hour standard, averaged over 8 hours, that's the one that's about to go to 0.075. There's actually real conversation about the ozone standard, pressing the EPA to put the ozone standard to 0.06 part per million ozone, which would, that will be interesting because there are a lot of communities that would have significant problems dealing with getting down to 0.06 on a regular basis. So what's the governmental hook for decreasing the ozone standard? Why should a state care that its ozone standard goes to 0.06 part per million or 0.075? What's the penalty? Well, yes, the penalty is money. Specifically, what kind of money? There you go, yeah. So all the nice roads that lead to the ozone pollution, or some of them, are in fact, so there's a pretty big stick that goes into it. The state of North Carolina, and I'm very familiar with this because of my seat as the physician member of the North Carolina EMC, is frankly, the EMC and your state government is very concerned about maintaining good air quality and maintaining good health. That's coupled with the interesting dichotomy of also maintaining, economic realities, and so it's a vexing issue that we have as a society. But anyway, this is the current ozone standard. So where does ozone come from? I suspect you all have heard this ad nauseum, but just for those of you who may not have, or for those who may listen to this tape who may not have, ozone derives from the reaction of ultraviolet light with volatile organic compounds and oxides of nitrogen. These things being generated by combustion, by combustion, usually of organic materials, fossil fuels, whether it be coal, whether it be oil, whether it be diesel, whether it be gasoline. In a particular region, this can be worsened by heat inversions, and in fact, the local geography can be very important in whether or not a particular community has lots of ozone exceedances. Because the perfect formula for having a lot of ozone in the summer is to be in a community ringed by mountains, have a heat inversion so the air is trapped, have a sunny day so you cook the VOCs and the NOx such that you make a lot of ozone that day. Cars don't make ozone. What cars make, or what chemical plants make, or what coal-fired power plants make are the precursors to ozone. And so it's generated fresh every day, but that's where it derives from. And I mentioned these are the points, these are the sources. Automobile and truck traffic is a big source. Point sources, which is basically any industrial site or power plant site, and these are both become pragmatic points of regulation. Other things that you don't think about, but actually are there. There's a lot of off-road vehicles. There's a lot of gasoline-powered tools. There's a lot of other things that can generate the fumes that contribute to the production of ozone. Okay. So what does ozone do to health? So this is a slide looking at the number of respiratory admissions to a hospital. This is a Canadian study relative to the 5-day average. This filtered refers to a statistical maneuver. 5-day average of the ozone concentration. This is expressed in parts per billion. So this is 40 parts per billion. This is 0.04 parts per million. This is 1/2 of the current ozone standard. And what you can see is once you get above 20 parts per billion, you can see a somewhat linear relationship between increased admissions to hospital, or the number of admissions to hospital, relative to the ozone concentration. And I show this slide. There are actually more robust slides at higher levels. The reason to show this slide is to make the point that even modest levels of ozone can have an effect on health outcomes within a community on an epidemiological scale. Now this slide, so I can say whatever I want to about this slide because you can't see this slide, but so the take-home point of this slide is this was a study. This represents a study that was done in looking at New England air and looking at particulate matter less than 2.5 microns in size and ozone. And this is now the current standard. This green line again is the ozone standard, and these spikes represent ozone events and exacerbations. And the take-home point is that at exacerbation levels, these spikes that are below this line are also statistically strongly associated with wheeze and chest tightness and persistent cough and shortness of breath. So again, this is another demonstration that pretty modest levels of ozone below the current standard are clearly linked or associated with exacerbations of disease. So I've gone from a couple of slides that show in an epidemiological basis what air pollutants can do. So these now we're getting into studies that we've done with volunteers to examine the effect of ozone on individuals that undergo an experimental exposure. So this was a study in which we used a fairly high level of ozone for a shorter period of time, and that's a way to mimic a day without needing a whole day to mimic it. And what will happen to a person in general when they're exposed to ozone? There are two major responses that occur. One response that happens fairly quickly, and a small number of people will have this in real life in a natural setting. There will be a sensation of difficulty in taking a full deep breath. That is a pain response that has nothing to do with inflammation. You can prevent that response with ibuprofen if it's done beforehand. You can intervene with that response with opiates, which are probably not the best thing to have on hand for an ozone exacerbation, but that was done experimentally to demonstrate that this immediate pain response, which feels like an inability to take a really deep, good breath, and that's what people will complain of, and that will go away. That event, though you can mimic it, is really not the driving force behind the public health offense. And so that's basically what all these these data represent, is that in our study, when we compared air versus ozone, we looked at forced vital capacity, which is all the air you can blow out in the forced maneuver, and the FEV1 is how much of the air got out in 1 second. Those are fairly standard measures of lung function, and basically what we found is that these two measures were both immediately decreased as a result of the ozone exposure, which is not terribly surprising, ozone compared to clean filtered air. What we also did is these volunteers, the morning after our experimental ozone exposure, agreed to undergo a bronchoscopy, where they did a procedure where we bronchoscoped them and we were able to get airway fluid from them. We analyzed the airway fluid for the amount of neutrophils, which are white blood cells that cause inflammation. And because they were allergic, we also looked at eosinophils. And the percentage of the total cells that were neutrophils and the percentage of total cells that were eosinophils were significantly increased in in these people after the ozone exposure. So this is direct evidence that in a person who's set up to be allergic, ozone can cause an increase that persists to the next day of neutrophils, and which probably are also involved in acute asthma worsenings, and eosinophils, which are the cells that are one of the defining biological features of allergic disease. We've taken the study one step further. Actually, we get paid to take the step several steps further. And we've actually also gone to a point where, because bronchoscopy gives us very reliable information, but there's a limited number of people who have the interest in being bronchoscoped at the end of a study. So we actually worked on how we can get sputum samples and go through a maneuver where you can induce sputum basically with high concentrations of salt water that loosen up the mucus, and then we coach them through a coughing maneuver, and then with the sputum we recover. We can do much of the same analyses that we can do with a bronchoscopy. What we did is we looked at monocytes and macrophages. These are cells that are present in all kinds of tissue, including the lung, that coordinate a lot of other responses. They also probably coordinate a repeat response to something you're allergic to. And basically, there are molecules on these cells that are really responsible for doing that, including a molecule called HLA-DR and CD86. These two molecules are very involved in coordinating how the body can take something you're allergic to and present it or give it to T lymphocytes. The T lymphocyte is the immune cell that controls almost all of the programmed immune response that you have. The T lymphocytes are the cells that if you get HIV disease are destroyed, okay? So they are really important in modulating all kinds of immunologic responses, including the ones that cause allergy. So let's say you're allergic to house dust mites. So what part of the house dust mite are you allergic to? You're allergic to mite exhaust is what I call it. So there's an enzyme in fecal matter that's actually very aerosolizable in house dust mites that you become allergic to. So when you inhale that and the molecule that's in that your body's responding to, a group of cells that include these monocytes and these macrophages will gobble that stuff up, digest it a bit, and then they'll put it on their cell surface and they'll wander over. Well, it's more complicated than that, but essentially they wander over and they come into direct contact. They kiss the T lymphocyte, and by doing that, they present antigen to the T lymphocyte that then says, "Wow, you got mite poop here, and we should respond to that." And that then causes a whole other generation of making all the other biological things that help to cause you to be more allergic. Basically, what this shows is that after an ozone exposure, compared to data we got either at baseline prior to any study or exposure to filtered air, which was a control, we found that after the ozone exposure, these innate immune cells that help coordinate response with the rest of the immune system have increased amounts of these molecules that are crucial in allowing the body to respond to the biological environment. If you remember that ozone not only causes pus to come into your lungs, which are the white blood cells, or eosinophils to come into the lungs if you are allergic, it will also cause cells that are there to surveil and protect you to be more revved up. So it makes you more likely to respond to things that you may be allergic to. That's a really important concept. Now, I'm showing you these data. These are data that we just very recently published, and this was the precursor to a study that we're literally working on with our EPA colleagues to publish looking at 0.06 part per million ozone. At the 0.08 part per million ozone, what we found is that if we looked at neutrophils and the number of macrophages in sputum, we didn't find much of an increase in macrophages. But we did find increase in neutrophils at this level of ozone. So this is actually one of the many bits of information that would convince the administrator that 0.08 was still maybe too high. The other thing we found is that when we did look at the macrophages, although they were not increased in number, some of these molecules, including the CD86 and the HLA-DR that we just talked about, were also increased. And these sputum samples were collected the day after the ozone exposure. So what happened was they underwent this experimental ozone exposure for 6.6 hours to mimic a workday. So if you're someone who has to work outside and you're exposed to this stuff all day long, and then we got sputum from them the next day. So again, modest level of ozone can cause pro-inflammatory or inflammatory effects in the lung. Okay. Now, let's talk a little bit more about how asthmatics might be more susceptible to ozone than non-asthmatics. So, so asthmatics and non-asthmatics can get pus in the lungs and can changes in the macrophages. But if you're an asthmatic, what happens? And these are data that actually they're in press. So what that means is that we got the paper accepted, but it still hasn't come. It's still being processed to come out in press. And so what we did is we compared with a fairly small number of people. We compared the responses to ozone in people who were normal volunteers, people who we called allergic rhinitis, but these are essentially people who were allergic but did not have asthma. And then these were people with mild allergic asthma they were allergic and they had asthma. And we looked at eosinophils and we looked at neutrophils, and we saw, we had a big variation, so we didn't see a significant increase in eosinophils, so there was a trend that there was more eosinophils in the asthmatics. That wasn't terribly surprising to us. We found actually this was modestly surprising. We didn't have quite enough to show a difference with the normal volunteers. Clearly there was a more defensible and routine response in the allergic individuals with regards to neutrophils. What was perhaps more interesting to us were some of the immune hormones that were made that really help modulate the downstream events. So part of the way that these macrophages and other cells cause the immune system to be activated is by secreting a whole bunch of things, and they're called cytokines. But a way to think about them, if you don't think about cytokines all the time, is that they're basically immunologic hormones. They're molecules that are secreted so one part of the immune system can cause changes in other parts of the immune system. And so IL-1 beta is a really important, very generic immune hormone that causes inflammation. And it was actually higher in asthmatics at baseline and much higher in asthmatics after ozone compared to these other populations. IL-6 is kind of the same sort of deal. Interleukin-8 is specifically involved with recruiting neutrophils to the airway, and interleukin-8 was significantly higher in asthmatics compared to the other groups. What was really intriguing was interleukin-10. So interleukin-10 is an immune hormone that's an off switch. So, a lot of these other things promote inflammation, but typically your body wants to shut it off when it's done with it. So interleukin-10 is an immune hormone that in certain circumstances turns off immunity or downregulates inflammatory responses. And we found that the asthmatics actually had very little IL-10 at baseline, and they certainly didn't produce IL-10 when they, after an ozone exposure. Now, the other thing you did with molecules that, unless you're an allergist, you're not likely to recognize, but are important. We looked at a bunch of molecules on some of these surface cells in the airway that are really important in allergy. Two that I'm going to point out are the Fc epsilon R1. So if you're on Jeopardy and you have to match up the Fc epsilon R1 to what's the receptor for the IgE molecule, that would be the answer. So now you're going to ask yourself, what's IgE? IgE is the body chemical you make when you become allergic to something. So when you have an allergy skin test, what they're really looking for is to see if your body has made IgE directed against whatever it is they looked at. And that's the reason you get a little welt there when you have your skin test. The take-home point here is that after an ozone exposure, if you're an allergic asthmatic, your macrophages, and in fact probably other cells as well, have an increased amount of the receptor that allows it to bind to the IgE, therefore bringing a molecule into the airway that helps you respond to something you're allergic to. Okay? The other one I'm going to point out is this thing called the TLR4, or the toll-like receptor 4. The take-home point here is that this is a molecule that helps you respond to bacterial endotoxin and all kinds of things without having an otherwise organized immune response. We also have done an experiment to find out if you are exposed to ozone, are you actually more reactive to the thing that you're allergic to? And this is the answer to that question. So when we expose individuals to ozone, and then we also challenge them with an inhalational challenge to something they're allergic to, we look at the amount of that material that's necessary to cause a very modest wheezing event. We had mild allergic asthmatics, and after an ozone challenge, we then had them inhale house dust mite, and then we looked at the amount of allergen. This is expressed in allergen units, but this is a way to measure mite allergen. And we found that there was more of it required after air than there was required after ozone to cause the wheezing effect. So this is, in an experimental setting, demonstrates directly that after exposure to ozone, you are more reactive to the thing that you're allergic to. And we also found this in the nose, where we looked, instead of looking at wheezing after inhaled allergen, we looked at eosinophils that were present after squirting at something you're allergic to in your nose. So this is artificial hay fever. But after ozone, there's more eosinophils in the nose that are caused by allergen than there is if you had a clean air. In all these studies, the people were randomized, so they didn't know what they were getting on that day. We didn't know what they were getting that day. So it wasn't like we were looking hard, but we weren't looking special hard because we didn't know which day was which. Okay. So I've made this summary all the way through, so I'll probably just move on. The major take-home point is that if you're an allergic person and you're exposed to ozone, you will be, for a little while, more allergic to the things that you are allergic to. A big question for all kinds of diseases. You've heard of the Human Genome Project, and everything's about genes these days. And it turns out that there are a couple of genes that are probably really important with regard to the response of people to ozone. And one of these genes that are actually identified in the cancer literature is the glutathione transferase M1 gene. Okay? So glutathione is a molecule that you need to prevent oxidant stress. Okay? So bleach And Clorox, your body makes bleach and it makes Clorox. And that's one way that it kills things. But when you have too much of it, it spills out and it causes injury. So you have to defend yourself against this oxidant stress. Ozone is in fact a direct oxidant. And many of the things, of the pollutants, when you inhale them, induce oxidant responses. And if you have too much of that oxidant response, if you make too much bleach-like molecules on your tissues, you hurt the tissues. So the way the body defends itself is to have molecules like the glutathione transferase M1 gene. So that's why we're going to focus on it in part because of that, and in another part because it turns out, for reasons that no one's really figured out, about 30 to 40% of the human population don't make this enzyme. They have genes that they're homozygous, so when they have two genes, both of those genes do not code, are such that you don't make the protein. About 30 to 40% of all human populations, regardless of location, ethnicity, gender, will be null for this gene. So they don't make this, okay? So, and because it's a big number, it's like easy for us to study because I don't have to screen 10,000 people to find the 10 that I want to look at. I can screen 10 people and find 4 that are going to be this way. Okay. And the biology of this is, basically, there's another molecule that regulates a whole bunch of these kind of things, but among the things it regulates is GSTM1, and if you're null, the GSTM1 null gene has been associated with increased response to ozone, increased response to diesel exhaust, increased response to tobacco smoke, and it's also been associated with increased risk for all kinds of cancers and other kinds of settings. So I'm going to stray from ozone just a minute because this is a really cool story. And it's not cool if you're the one affected, but it's a really, really neat story. This is a study that was done in Southern California by a group that has access to a big population of kids. And what they did is they went through and they said, so what is the risk of smoking during pregnancy to subsequent respiratory disease in children during the toddler years? So what they did is they found a group as a reference group. They said, okay, our hypothesis is that if you're a GSTM1 null, you're going to be at higher risk for wheezing disease. So as their reference group, they said, okay, I'm going to find women who did not smoke during pregnancy, and the kids were GSTM1 sufficient. They actually make the enzyme, they're fine, okay? So everything was compared to that group. And so they looked at, I mean, you know the punchline before I get there, because you can see the really big thing up here, okay? All right? So these are GSTM1 sufficient individuals born to mothers who didn't smoke, all right? These were GSTM1 null individuals born to mothers who didn't smoke. These are GSTM1 sufficient individuals born to mothers who smoked like chimneys. And these are people who were GSTM1 null and their mother smoked during pregnancy. And what this was the risk of having an ER visit for wheezing before the age of 5. Okay? So your risk for wheezing disease is directly associated with both the environment, in this case it was the mother smoking, and the genetic ability to respond to that environment, which in this case was the glutathione transferase M1. There are other genes that are important in this as well. I'm going to tell the GSTM1 story. Okay? So with that backdrop, I'm going to tell you a little bit about a study that was done in Mexico City. There's a woman named Isabelle Romeo, that's a French name, which I mispronounced because I don't speak French very well, but she has worked for years in Mexico City at kind of the Mexican version of the National Institutes of Health. And they have found a cohort of children with asthma, and so their question was whether or not children with asthma who were GSTM1 null had more ozone-related health outcomes related to those who were GSTM1 sufficient. And I would try to walk you through this table, but the real punchline here is that the GSTM1, or else I wouldn't have shown it, is that the GSTM1 null children really are the ones who have, when they looked at the whole population, there was clearly an effect of ozone in causing wheezing events in these kids. But when you segregated on the basis of the gene, the null kids were really the ones that drove that finding. GSTM1 null children had increased exacerbations of asthma that were occurring 24 to 48 hours after a naturally occurring increase in ozone in Mexico City, which, by the way, has a huge ozone problem because it's ringed by mountains, lots of sunlight, lots of people. It's actually the most populous city in the world, the metropolitan area of Mexico, which means, and they have lots of combustion of all kinds of stuff. Okay, so I've shown you the smoking story and then one of a number of examples about how GSTM1's been shown in real life settings and other settings. Now these are our data. So our question was, does GSTM1 modulate the experimental response to ozone? And we looked at effect on lung function, this pain response, and GSTM1 didn't have any effect on the pain response. Both are GSTM1 sufficient and negative people wheezed or had, had temporary decreased lung function after ozone exposure, which is typical, but there was no segregation of that effect on the basis of the gene. When we looked at pus, however, or we looked at neutrophilia in the lungs and macrophages in the lungs, and this is cleverly circled because we really were looking at the day later because that's the time point that meshes best with the epidemiology. What we find is a day after an ozone challenge, GSTM1 null individuals have a significantly increased amount of neutrophilia present in the airways than compared to the GSTM1 sufficient individuals. And likewise, when we look at activity of the pro-inflammatory activity of the cells, the ability of cells to generate bleach themselves was actually augmented more in the GSTM1 null people. Their ability, the phagocytosis capability, they were more activated, and the expression of this HLA-DR, which is the molecule that helps you, enhance your immune response. All those things were elevated after ozone in the GSTM1 null individuals. So thus far, what I hope you've heard is, this is what I meant to say, was that ozone causes a problem. There's epidemiological data that it causes problems, and it causes problems at fairly low levels. We can experimentally study this in volunteers, and we experimentally study it in volunteers. It augments all kinds of parts of the immune response, including those parts that make you allergic, okay? We've also talked a little bit about a really commonly occurring gene that in some people, in a sizable number of people, may enhance their susceptibility to the effect of ozone. Now I wanted to get a little bit about some of the more global things that might happen, and this, I'm sure you all have seen some iteration of this or another. This is the average temperature, and this is going from 1880. This is above a mean, and the take-home point here is that across the past 100 and some years, or 108 years, there's been a significant increase in the mean temperature found on the Earth. And so this gets at the global climate change. Whether you believe it's anthropomorphic or whether you believe it's natural, I'm not even going to quibble that point. The fact that it occurs, I mean, I can take a thermometer, I can take your temperature, I can tell you have a fever, whether you are exercising too much or whether you're sick, okay? And so the temperature has in fact increased, okay? Now, these are projections of what that climate change and an increased heat, which therefore means more heat inversions and more other events that will promote ozone. These are modeled projections of what this might mean to the increase in the maximum one-day average of ozone in a variety of areas. And you can see that each of these things that have a dot have at least some degree of a change, but some areas, including our areas, will have an 8 to 12% increase in the number of ozone exceedances, and likewise, in some areas you have really big exceedances. This isn't because they're hotter, but this is because they have more people and more cars, at least at the moment. Now this slide, which you can, if you can get it, if they can provide it to you, you can look at it at your leisure, but this is just kind of breaking it down by specific cities, both the changes in the 8-hour and the 1-hour ozone level using the old 1-hour standard. And the take-home point here is that by the 2050, if the projections are correct, there's going to be a lot more days with ozone that are out of compliance with those current standards. Now, because I'm an allergist and because I've talked a little bit about how ozone can augment response to allergy, I'm also going to tell you that global climate change may result in more allergy as well, and specifically what it can do, and I'm going to really draw your attention to this. What these data show is the amount of the material in ragweed pollen that you become allergic to. Okay, there's a molecule in ragweed that that's the stuff that you become allergic to if you hate ragweed. And with global climate change, what's going to happen is that the amount of that material per pollen plant or per plant is going to be remarkably increased. These data derive from an experiment in which ragweed was grown in 280, 370, and 600 micromoles per cubic meter air of CO2. So this was experimental CO2 increase due to global warming, and the CO2 directly drove the amount of ragweed pollen, or the amount of ragweed allergen that was in the pollen. Furthermore, if you look at actual pollen levels and ragweed growth, what you find is actually higher in suburban and urban areas and it's a function of both CO2 and temperature. So you think of ragweed as being something out in the country, but in fact, because of street canyons and because of where we are, there's actually more ragweed pollen and present in cities because the atmospheres are actually friendly to it, and because of the increased CO2, we can predict that ragweed will be more potent. Okay. So now what could we possibly do about this? Now, one thing we could do, and I think I may have a slide that speaks to it, is that if you have asthma, better baseline control of your asthma will actually be really helpful to you. We've shown that if you, and I think I'll show you a slide in a minute, but that if you take inhaled corticosteroid, which is one of the cardinal key treatments for routine control of asthma, and you use it fairly regularly before an ozone challenge, your response to ozone is much less. So if you decrease your baseline allergic disease, that's important. That's important for any number of reasons, and that's probably the thing that. How many of you take care of people with asthma? So how many of you have patients that I quit taking it, or they took it wrong? I mean, using an inhaler is hard. I mean, it's really well, you didn't use your inhaler. Well, what do you have to do? You have to learn a lot of stuff. I mean, you have to juggle, literally. You juggle with your, timing the spray that comes out of the tube at about 80 miles an hour so that you're inhaling instead of eating it is actually a trick. So it's real easy to not do it well. But if you do it well, if you do use the corticosteroids beforehand, it'll be helpful. But there are other molecules that are kind of useful as well. Now, when we talk about antioxidants, we really talk about two basic kinds of chemicals or two kinds of changes. One are the vitamins that most of us are used to thinking about, and these are basically antioxidant sponges. These are small molecular weight molecules that basically just absorb bleach or bleach-like molecules. They directly are sponges, and they can be found inside a cell or can be found in the cell in the surface, but they absorb things. The other kinds of things that can happen is that you can upregulate this NRF2 that would upregulate all kinds of enzymes that also help us deal with oxidant stress. So people have looked at ascorbate and glutathione and a variety of other micronutrients, actually uric acid, which is made by the body, and although you think of it as being part of gout, it in fact is it is an antioxidant, and various forms of vitamin E. And then there are these other things. I'm going to touch on ascorbate and vitamin E, and I'm also going to touch on sulforaphane here in terms of antioxidants. So this was a study, this gets back to the kids from Mexico City. So one of the things they decided to look at is whether or not oral antioxidant therapy with alpha-tocopherol, which is a form of vitamin E, and ascorbate, which is vitamin C, would mitigate the effect of ozone. And so what they looked at, and actually this blue probably could have projected better. I was told it would project better, but I didn't have the time to actually change the color of the slides, so I apologize. But so this looked at peak flow, this looked at FEV1, this looked at peak flow. This is the most interesting slide. So they looked at these kids, and they looked at kids who were on placebo and then kids who were on the combination of vitamin E and vitamin C. And this is decrease in lung function following the ozone exposure. You can see on placebo, they had much worse of a decrease in lung function compared to when they were on the vitamin supplementation. And they also got nasal lavage from these people looking at some of these inflammatory hormones, and basically what you can see is that the change per increase in amount of ozone in the air was essentially done away with if they were on the supplements compared to when they were on the placebo. Now, what the slide I'm not showing you, but is actually true, is that when you further subdivide this, what's very clear is that the people who benefited from the antioxidants were the GSTM1 null individuals in this population. Okay. So the other molecule that we're actively studying, and a lot of other people are studying, you could actually buy it at one of these highfalutin, I don't know which highfalutin grocery stores here, it's this Whole Foods where we are, up the road, but it's sulforaphane. Now you don't buy, what you buy are Braco sprouts. They're actually the flowering parts of the broccoli plant. It's not the typical broccoli, it's the Braco sprouts. You can buy those, and the ends of those are actually very, very rich in sulforaphane, which is a molecule that upregulates NRF2. And what this slide shows is compares the amount of a particular, of enzymes that are present in nasal tissues with different doses of sulforaphane that were given, in by way of eating Braco sprouts. And the take-home point is that you really can increase your antioxidants if you eat the Braco sprouts because you're eating the sulforaphane. And there are all kinds of attempts to make it more medicinal so you don't actually have to eat the Braco sprouts. So we're doing a study now where we're asking people to eat Braco sprouts before they're exposed to diesel exhaust because actually diesel is a really pro-oxidant thing. So there actually is an FDA rule. We had to get approval. And the biggest side effect from eating Brussels sprouts is flatulence. And so that's about the only side effect, which if you're trying to get your kids to eat it, might actually be a reason to get them to eat it. So you're treating one aspect of air pollution by generating another aspect of air pollution. But there's very real interest, and there's evidence to suspect, and actually this is also really important. This really derived from cancer risk, because oxygen stress at a low level in all kinds of places, including when you eat charred meat, the risk of colon cancer. The charred meat basically is the home of all kinds of pro-oxidant molecules. And so there's a lot of interest in this in the cancer world as well, and that's where a lot of this GSTM1 stuff derived, and then we extrapolated it to what we're doing now. So, yes, antioxidants are still under investigation, but they may be useful. They don't replace traditional therapies for asthma, and there may be some groups that are more susceptible to others. And once we really, really confirm that, one could imagine the day where you could go and find out if you're a GSTM1 null or not. And then if you are, then you might want to do other things. One of those things may not be to call your insurance company and tell you you're GSTM1 null, but although that's like a third of the population. Now, what can we do? What can individual patients do, and what can communities do? Okay, so one thing you can do, and this sounds trite, but decrease tobacco use. Tobacco use trumps everything. If you've got a smoke in your house, frankly, all the air pollution, orange zone things don't mean a bit of hooey. Because I actually shouldn't say it. It probably is worse for smokers, but tobacco smoke is, indoor tobacco smoke gives you just all kinds of particulates and everything else. So limiting tobacco use is a big deal. Local environmental controls inside your house. So what do you do inside your house for ozone? There's not a lot you realistically can do inside your house for ozone. There's a lot you can do for particulates, and ironically, it's keeping your windows closed, which sounds counterintuitive on a springy day, is really true for pollen. The other thing you really can use is the every 3-month air filters that you put in actually really are a little bit better at trapping the things, if you remember to change them. That's the caveat. Because inside houses for allergy, the other big thing, so you might have guessed, I think is kind of important, is humidity control. Air conditioning is actually a pretty good way to control indoor air pollutants, and specifically controlling biological things that promote allergy. Allergen levels, house dust mites require humidity, and so humidity control, decreasing humidity is a big deal. And ideally, you should have it around 50 or 40%, which is not easy to do, but it is doable if you've got patients that really need that. But indoor environmental controls. The other thing is living away from a roadway, and that's because of both ozone and particulate pollutants. If you live within 500 meters of a road, you are much more likely to have all kinds of bad things happen to you. Maybe all kinds of bad things happen to you, but specifically things because of the air you breathe. And we've not touched on at all the role of ozone, and more importantly, the role of particulates in myocardial infarction and stroke, which are very real. All those things happen closer to a road, likely in part has been statistically associated with particulate load, and particulates are a whole different deal. But ozone's also close to the road, and then there are the other things that we could do. All right. The social experiment that was done in 1996 in Atlanta was to clean the air for the Atlanta Olympics. And the motivation for this was so marathoners would not drop out of the race and set world records. I'm not kidding. That was the motivation for this. It wasn't the health of the population of Atlanta. That was a convenient side effect for a couple weeks. And that was the big deal in Beijing. You remember the Beijing Olympics, where if your license plate ended in a certain numeral, you couldn't drive, and if it was too smoky, you wouldn't drive at all. And in fairness, the Chinese did a pretty good job, given where they're starting from, cleaning up their air for the Olympics. But these events prove that you can actually do something about the air pollution. Okay, so what this slide shows is the percent of the percent of the NAAQS standard that was present for all these NAAQS. And the take-home point here is that the one that was really the most significantly, when you looked at the pre-Olympic period versus the Olympic period versus what happened after the Olympics were over and they reopened the roads and people could drive willy-nilly again, what you found is you found a very significant dip in the ozone level, okay? And so certainly you can have a decrease in ozone. And was there an effect on health outcomes? So in medicine, at least in healthcare systems, and I work for one, my several bosses will slap me around when I get home, but the best data you can get is billing data. And so what they looked at were Georgia Medicaid claims, various economic claims for health maintenance organizations, visits to EDs, and the Georgia Hospital Discharge Database. And what they did is they looked at the baseline period and the Olympic period, and they looked at the percent change in those hospitalizations associated for asthma events, and they all decreased. Some decreased up to 40%. To be sure that it wasn't simply that people weren't going to the ER at all because it was too hard to get to the hospital, they looked at non-asthma events as well. They didn't really change, but the asthma-specific events actually had a significant decrease during the Olympic period. So this is kind of cool. Now, this is far from proving mechanism, but is a very interesting association, and I think it demonstrates that if communities decide they're going to change traffic patterns or change ways that the kind of fuels they use, and that's one reason why there's so much interest in higher mileage, various different kinds of fueled cars. The data, I think, are pretty compelling that if you do things that decrease ozone, you will probably do things that improve health outcomes. Okay. The last thing, this is our endotoxin study, but we actually showed the same thing with ozone, is that is if people use their steroids prior to an experimental challenge, they have less pus and they actually have less changes in these immunologic cells as well. And this is our ozone data looking at the effect of placebo versus different doses of the drug called fluticasone. And what we find is that the percent of total cells that are neutrophils, if you look at the mean, they decrease after ozone challenge in the people who had fluticasone. And so with that, I've finished going through my slides. The kind of work we do can't be done without all kinds of people, and I could read all these names, but I'll let you read the names, but suffice it to say that I have a really, really good, the people in blue are people that actually work in our center or used to work in our center that actually did the work that I asked them to do. These people in green are our EPA colleagues, and Steve Kleberger works at the National Institute for Environmental Health Sciences. With that, I really appreciate your attention, and I hope I didn't go too quickly. Thank you very much.